So what happens when we take a probiotic? So these are ATCC, this cell line HT29. These are human colonic epithelial cells, and when they were administered Bifidobacterium longum, by 60 minutes they start colonizing. They multiply by 120 minutes, they increase, and that's how they increase in their number in the intestine exponentially. And it's not only about increasing, it also get adhered. So if you see this, this is the intestinal villi, and this dot is a colloid gold electron microscope. And that labeling was done with one of the antigens of Lactobacillus casei. So when Lactobacillus casei with this labeled gold was given to the, this is a mouse study, you can see the organism, part of the organisms within the epithelial intestinal mucosal colocyte, and they also entered into the villi, into the intervillous crypt space. So they get adhered to the intestinal epithelial cells. So again, the goal of these probiotics are to return back the normal function. So this is a complex set. Let's make it simple. So if we, sorry about my throat. So we can divide the function of probiotics into two broad categories. One, it could be immunologic benefit. One, it could be non-immunologic benefit. So in the immunologic benefit, there is activation of the local immune system. They increase local macrophages so that the antigens are presented to the beta cells. There is increase in secretory IgA production from the intestinal lumen. There is modulation of cytokine profiles. Dr. Ghoshal had already said about the different cytokines. And there is induction of tolerance to food antigen. And in the non-immunologic group of functions, they compete with the pathogens for nutrition. So they would proliferate very rapidly, and they'll compete with the pathogens for nutrition. So the pathogens get depleted and they start dying out. And they could alter the local pH to create an adverse environment to the pathogens. There could be bacteriocin production. They can scavenge superoxide radicals. So lots of functions have been believed to be present with these probiotics. So let's look at some of the indications. So there has been several studies now, and all these studies like acute diarrhea, antibiotic-associated diarrhea, Clostridium difficile diarrhea, H. pylori, hepatic encephalopathy, you heard about that, non-alcoholic fatty liver disease, irritable bowel syndrome, all these are indications where probiotics have been studied. Then if we look at the level of evidence, as scientists, you would always believe in evidence, right? And as clinicians, we would always look at evidence before we prescribe the drug to the patient. So when I talk about level 1 evidence, it means like these are systematic review or meta-analysis. Level 2 evidence are either randomized study or large case series, cohort studies with a large benefit. And evidence like 3, 4 are just case series or individual case reports. So if we see in only one indication, like antibiotic-associated diarrhea, the level of evidence is very strong, 1. And most of the indications, it's 2. And then in inflammatory bowel disease, something called pouchitis, the evidence is 1 to 2. So really, the level of evidence is not very strong for all indications. So the indication specifically which it could be beneficial is antibiotic-associated diarrhea. Now, when we look at probiotics, it has to fulfill certain fundamental criteria. So obviously, one, it has to be safety. So it has to be from a human origin. It should be non-pathogenic, and it should be resistant to antibiotics. Otherwise, the antibiotic that you take will kill the organism. So it has to have some functional criteria, the resistance of against acid and bile condition. And it has to adhere to the mucosal surface that I showed in one of the previous slides. And it has to be having a clinically validated and documented health benefit. And then there should be lots of physiological criteria that it should have antagonism against enteric pathogen, lactose tolerance, cholesterol assimilation, and technological like genetic stability. It should be genetically stable. It should have, it should be resistant to bacteriophages, and it should be amenable to large-scale production. But when we look at the actual journey of probiotics from the production house to the consumption, when it goes to your gut, it goes through a very long process, very complicated steps. Like production may be perfect, it may have the right kind of organism, it may have the right proportion of organism, physiologically it is good. But while processing, packaging, storing, it has to maintain a cold chain. So all these things might happen, like oxidative stress, osmotic stress, during the processing and packaging. And when you take it, it depends what is your resident bacteria. If we don't know about the resident bacteria, we don't know the interaction between the probiotic and the bacteria. So all these things could actually could result in a very low quality probiotic that you take. It may have been good at production, but by the time it reaches your intestine, it may lose many of its property, many of them may die. And then now there are studies actually from both India and West that have shown that whatever is written on the label, actually the main probiotic do not contain that, or the quality is poor, the concentration is poor. So that's something of concern. So this was mostly about the bacteria-based probiotic. So can we circumvent that? So if we use yeast, because yeast also has several probiotic properties, it has immunomodulatory properties. So there has been many yeast that has been tried, but only Saccharomyces boulardii has been shown to have some probiotic effects. So this is an electron microscopic image of Saccharomyces boulardii grown on this human colonic cell line, and this is Salmonella typhimurium. So what is the benefit? If you compare to bacteria, yeast is not usually present, Saccharomyces is not usually present in the gut, whereas bacteria are like 99% of the organisms inside the intestine are bacteria. So the interaction between bacteria and bacteria would be different from interaction between yeast and bacteria. Yeast are 10 times larger than the bacteria, so there is a steric hindrance. So the bacteria would compete for space with the yeast. So that gives yeast a better advantage of growth. The cellular properties of bacteria and yeast are different. So the immune responses are different. Like for bacteria, it is like toll-like receptor. Again, you heard from Dr. Ghoshal. And from yeast, it's more of lectin-based receptors. And if you see here, the yeast would survive at a wider range of pH and at a higher pH. And it also can survive at a higher temperature. And intestine has different temperature and different pH at different levels. Like stomach has different, duodenum up to the colon, the pH level increases, decreases. So the probability that yeast would survive the entire intestinal transit is much higher than the bacteria. And of course, yeast won't have antibiotic resistance, and there is a theoretical possibility that the probiotic bacteria could have some horizontal gene transfer of resistance genes, which may be harmful for the patient in the long run. So if we talk about the adverse effect, I guess no one who had taken probiotics got any adverse effect. So there are some documented adverse effects like GI side effects. These are minor effects actually. Could be cramping, nausea, altered taste. But potentially serious toxicities have been reported. So there are more than 30 cases of fungemia, like fungemia with Saccharomyces boulardii, several cases of bacteremia and sepsis associated with lactobacillus. How did I know this? Because when this patient had sepsis, blood culture was done. So the organism that grew in blood culture was same as the organism they took in the probiotic preparation. Right? So there were reports of endocarditis associated with lactobacillus. So there could be some deleterious metabolic effects. So there was a very nice study published in the Lancet like around 7-8 years back from the Dutch pancreatitis research group that was called the ProPatriot trial. So what they did was, patient who had severe acute pancreatitis, one group they treated the patients with a probiotic preparation, then the other group they did not give probiotics. And what was not expected was more patients who was treated with probiotics had died actually, which means that the probiotic was associated with higher adverse event or mortality in that group of patients. So that shows a deleterious metabolic effect of probiotics. And there could be some potential adverse events like excessive immune stimulation in susceptible individuals if the patient have some immune overactivation, the gut probiotic may aggravated immune activation or potential gene transfer. But these are theoretical side effects. So far, there has not been any reports of those events. So when you go to the market and buy probiotic next time, you should always see, because one thing is very important, these probiotics are very much strain specific, like not even species specific, but even strain specific. So you have to, the companies have to list few important things like, I'm not sure if you'll get everything of these, but at least you should look at the genus and species identification, especially the number, the strain designation. There should be a viable count written on the package. Recommended storage conditions has to be. So these top three things has to be there in a packet. Only then the probiotic could be believed to be providing a health benefit. So the caveats of probiotics are only select microorganisms are available as probiotics, and 30%, 70% of the gut organisms are unculturable. So when we give a probiotic, we do not know what the crosstalk between the probiotic and the host or microbes will have. We don't know the previous situation of the host microbes, so probably many of the probiotic effect of the preparations could be nullified. We do not know we do not know about the effects of resident microbiota. Okay, so a single probiotic do not fit all.